So Unit 4 is going to focus on cell communication and tying that in also into the cell cycle. So we're going to start with the basics of cell communication and we'll move on from there. So cells are going to communicate basically two ways. They're going to communicate via direct communication, so one cell actually touching another cell, so cell-to-cell contact, and we'll look at some specific examples of that. Or they're going to talk to each other over a distance, and that can be a short distance with our local regulators, or it can be a long distance, which is what our hormones do when they are produced in the gland and they travel a long distance through the bloodstream to the target cell. So we're going to look at all of these in detail, short distance, long distance, direct communication, with some specific examples that you can use to describe these different kinds of communication. So starting with direct contact. So like I said, that is exactly what it sounds like. The cells are actually in contact with one another. They are touching each other. So the first example we're going to look at here is with immune cells. So this is within your body, and these are our immune cells that come in contact with our own cells and with foreign cells. And the first example we're going to look at here is APCs. So APCs are antigen presenting cells. And so these are cells that will present pieces of an antigen. So an antigen is a foreign particle. So we can see this happening over here on the right. So I have this red here, the virus, that's my antigen. So that's my foreign particle. And this larger macrophage here, the big large yellow cell, so that large macrophage is my APC. It's my antigen presenting cell. So that large macrophage is going to consume the foreign invader. It's going to break it down, and then you'll see that it's putting pieces of the foreign invader out on the outside of the macrophage. So it's exposing pieces of that foreign invader out to other cells in your body. And so these other cells in your body, in particular this helper T cell here, so this helper T cell has to come into direct contact with the APC. It can't be nearby. The antigen presenting cell can't just show it to it and then the helper T cell knows that that's bad. It has to physically touch it. It has to come in direct contact with the antigen presenting cell. And so when this helper T cell comes in direct contact with the APC, then the helper T cell learns about the foreign invader. And so it learns about the foreign invader. It learns what it looks like, what its components are, so that as you can, if you follow the arrow here, it can then tell other cells in your body to help fight it off. Okay, it can tell B cells to make antibodies. It can tell killer T cells to get turned on and to go attack the foreign invader. But that would be one example of a form of direct contact. The antigen presenting cell has to come physically in contact with the helper T cell. You can see that happening right here between the two. They physically come in contact. The helper T cell learns about the antigen and then can go activate other cells. So another example is another immune example here with these killer T cells. So we said that one of the things that helper T cells could do is go activate killer T cells. So killer T cells are also called cytotoxic T cells, right? Cyto is cell, toxic would be poison. So these are cells that destroy infected cells. So you can see that happening in the second part of this diagram over here on the right. So here's my infected cell. Okay, and so this is our own cell. But you can see it has the virus in it. The cell is already infected. So the cell is done for. So the killer T cell or the cytotoxic T cell is going to destroy this infected cell. There's no reason for that cell to stay around. It's infected now, so it's not working properly. And if we allow it to stay, it will then just make more viruses. However, for the killer T cell or the cytotoxic T cell to kill the infected cell, it has to come in direct contact with it. Okay, so it had to come in direct contact with the helper T cell so it could be activated. And then it has to come into direct contact with the infected cell so it can destroy the infected cell. That's how it's going to know. Coming in contact with the helper T cell helped it learn what the foreign invader was, and then coming in direct contact with the infected cell helps it realize that that cell is actually infected. These cells can also help destroy cancer-forming cells because cancer cells produce, you'll notice here in the diagram here, the MHC proteins. These MHC proteins are what are displaying these antigens. And so cancer cells produce different MHCs from our normal cells. And so when the cytotoxic T cell comes in contact with that cancer cell, it can realize that that MHC, the piece that sticks off here, that MHC protein is wrong, and so it will destroy it. But again, it has to come in direct contact with that. Another example happens in plants. So the example that we have here is what's called a plasmodesmata. Okay, and so this is another example of direct cell-to-cell communication. And so what you'll see here is between these plant cells, there's these little holes, okay, these little openings, these little pores. And so the plasmodesmata is the opening or the pore there. And it's an opening in the plant cell wall. And so what that will do is it allows for actual cytoplasm to be exchanged between the two cells. And so that cytoplasm or cytosol exchange there allows for the chemical environments between the two cells to be connected. So we have direct contact between the two cells and direct exchange of information between the two cells because of this cytoplasm cytosol exchange. Water can move easily from cell to cell. Solutes can move easily from cell to cell. Depending upon the size of the protein, it can easily move cell to cell. And so that's a specialized thing that plants will have. They'll have this plasmodesmata for that cell-to-cell communication. Okay, so now that we've looked at some examples of direct cell-to-cell communication, let's take a look at short-distance communication. So short-distance communication is also called paracrine communication. So not the endocrine that you're used to with hormones. That'll come up later with long distance. This is short distance. This is from a neuron to a muscle cell or from a neuron to another neuron. This is cells that are really close together, but they're just not physically touching. Okay? So these are going to be what are considered local regulators. So they're helping control things and monitor things and alter things locally, nearby. Okay? And so their target cell, that's the cell that is being communicated with. Okay? So the target cell is going to be very close. Okay, so let's look at the first example here with these neurotransmitters. This is our diagram over here on the right. Okay, so the bluish-greenish part here, that's my neuron. And so that is the axon part of my neuron. And if y'all remember, axons carry messages away from the neuron cell body. So there would be the actual cell would be way up here. The message would travel down that axon towards the cell it's trying to communicate with, another neuron, muscle, whatever it is. So the neurotransmitters, which are the actual little molecules that come out of the neurotransmitters are released from the axon. So the electrical message travels down the axon and it releases the neurotransmitter. And the neurotransmitter is going to travel across this little space. The space here is called the synaptic cleft. This whole thing here is a synapse. And so the synaptic cleft is the little space between the neuron and the target cell. Again, muscle cell, another neuron, whatever it may be. So these neurotransmitters are going to travel across the synaptic cleft to the receiving end on the receiving cell on the other side. And there'll be a particular receptor on the other side. And so that receptor is going to be specific, kind of like enzymes were specific. That receptor will be specific for that particular neurotransmitter. And so that receptor will receive the neurotransmitter, and that will then initiate the communication, the signal into the target cell. That will trigger muscle contraction. It will trigger another message on another neuron. Okay? The receptor is what's key here, though. Because these neurotransmitters are traveling, they need to be sure that when they come in contact, they're coming in contact with the appropriate cell, with the target cell. You'll notice this synaptic cleft is not like sealed off over on the edges. Some of these neurotransmitters could, float out to the sides and go and come in contact with other cells. And we keep them from triggering other cells by having this specific receptor on the target cell. So that's going to be important. Okay? The binding to the receptor is what's going to help initiate communication on the target cell, okay? And it's going to be what makes them target specific. So another example here would be what we call quorum sensing in bacteria. So bacteria are unicellular organisms, but they usually live together in colonies. Okay? And those colonies will function similar in some ways to a multicellular organism. They will make changes and they will function together as a unit in some ways. And that's going to come from chemicals that are released from the bacteria. Okay? And so you can see here in the first little petri dish, I have a low density of bacterial cells. So they're releasing these chemicals, but they're not really getting to other bacteria. Okay? So I'm not getting, they're not talking to each other too much. Well, now, in the second one here, I get this high density. So in the high density, I've got lots of bacteria in the area, and there's lots of these chemical signals that are being released. And these bacteria are receiving that information. They're talking to each other. They are functioning. They can adjust their gene expression based on the information that they're getting. Maybe they need to stop reproducing because there's not going to be enough food because there's too many of them there. Okay? Maybe they need to secrete a particular chemical that would help protect them from something. Okay? And so in the high density region, these bacteria are talking to each other with these various chemicals, and that is influencing gene expression. Okay? We will focus a little bit more on this example in class, okay? And I think that will help. So let's look at our long distance communication here, our endocrine. So I know it seems like you're not getting really information. You just got three definitions: direct cell to cell contact, they have to touch each other; paracrine, local communication; endocrine, long distance communication. But you need to keep in mind that for a lot of these things, you need to have examples that you can write about, examples that you could use to help explain something. That's why we went through a plasmodesmata or an antigen presenting cell, okay, or a neurotransmitter. So do make sure that you are taking notes over these examples, because you will need to write about or use examples to help you write and explain. So the last thing we're going to look at here is endocrine communication, which is long distance communication. So this is happening in multicellular organisms. This is when your brain makes a hormone and it goes down and talks to cells in your toes. Okay, so this is long distance communication. Hormones, animal hormones in particular, are the example that we are going to look at here. So animal hormones are released from endocrine glands and they go straight into the bloodstream. Okay, endocrine glands, they have no duct. So they secrete their products right into the bloodstream, and they travel through the bloodstream to the target cell. And that target cell is going to be far away. And once again, that target cell is going to have a particular receptor on it, which is what allows the hormone to know that it has reached its target. That's why insulin isn't affecting, it's traveling through your bloodstream, but it's not affecting every kind of cell that you have, because it's looking for certain cells, certain receptors that are on certain cells. So we're going to go ahead and we're going to use that example here of insulin. So insulin is released in the pancreas. It's released from beta cells in the pancreas. And that insulin hormone is going to be released straight into the bloodstream, and it's going to travel and look for particular target cells. And the target cells, the main target cells for insulin, is going to be liver cells, skeletal muscle cells. These are going to be the kinds of cells that are going to be able to store extra sugar. And insulin's job is to decrease blood sugar. And it decreases it by telling these cells, that are able to store excess sugar, like the liver or the skeletal muscle, by telling them to pull the glucose out of the bloodstream. So the insulin is made in the pancreas, which is in your abdomen, near your stomach, kind of the upper middle part of your abdomen. So it's made there in the middle of your abdomen. It travels throughout your body, throughout the bloodstream, and in your liver, in skeletal muscles, so skeletal muscles found all over your body, it will activate those receptors. Remember, the receptors are what's going to initiate the cell's response. Well, the cell's response is to open up channels that allow glucose to come out of the bloodstream and into the cells, and therefore it lowers the blood sugar level as a whole. So these are kind of our basics of cell communication: direct communication, local communication, and long distance communication. And we're going to look a little bit more in depth now at how the cell actually kind of processes this information.